Hello, am I audible? Okay, thank you for confirming. Please let me know if you can see the video and if you can see the presentation clearly. Let me know if the video and presentation is visible to you. Okay, thank you so much for confirming. Good evening. Please wait till other students join, then we will start today's session. We will start at sharp 6 p.m. So kindly wait till then and let me know if there is any issue with the video or the with the presentation or with my voice. I think I will wait for a minute, then I will start with the introductions. Okay, meanwhile, when more students will join, they will eventually understand the things. Let us wait for 2 to 3 more minutes and then let us start today's session. Okay, so let me start by introducing myself. I hope eventually more students will join. So let us start. I hope my voice is audible, and I also hope that the presentation is visible. If not, kindly let me know. Okay. So very good evening to the student who have joined. My name is Mousumi Deb, and I am a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I'm your TA for this NPTEL course of Organic Chemistry in Biology and Drug Development, and today it is our week 3 live session. So basically what we do in every live session, we solve that particular week's course content. Like today it is the week 3 live session, so today we will discuss the course content which are in the week 3 of this course of organic chemistry in biology and drug development. Already the contents were posted in the previous week or on the previous Friday, and also you are given an assignment related to the week 3 course content, which is due in a few days. Okay, so today what we will do is we will discuss around 20 questions related to the week 3 course content. All the questions are MCQs like you have in your assignments, and also although the questions are MCQ, the answers I will be discussing in detail, so that if whenever the subjective questions are given to you in the final examination related to the particular topic, so Then what we will do is you can easily write the questions. Okay, so before starting the session, let me clarify a few points. So there will be a problem solving session every week, and the sessions will be on every Friday from 6 to 8 p.m. These sessions are live on Google Meet platform with the particular this is the link for the class, that this particular link you have used to for joining today's class, and this link will remain same for our entire course. Okay, so this you can write it down at any place, or you will find it in the PPT which I share already. And the important thing is, if there is any network disruption from my side, please do not leave the session immediately. I will try to rejoin as soon as possible. And if there is any issue from your side, you can rejoin at any time to the class. Okay, I am recording the session, that is why I have muted your microphones and cameras, so that I can record the session seamlessly. But the chat box is always open, and I am always monitoring the chat box. So if you have any doubt or any query, you can let me know through the chat box. And also, since I'm recording the session, the recording I will be sharing to you after the class, so that the students who have missed the class, they can go through the lecture, and they can easily find their doubts clear. Okay, if the doubts still persist after class, you can drop an email to me. I will share my email ID at the end of the session, and via email also I will answer your query if it is possible. Otherwise, I will answer your query whatever you have asked via email in the next class. Okay, if the answering takes a long time, I will discuss it on the next class. So as I have already mentioned, that this presentation and class recordings, it will be shared to you, and where you will find this presentation and class recording. So you have to go to the SWAYAM portal. I hope the SWAYAM portal is visible to you. So this is the announcement section of the SWAYAM portal, where the course content, everything is announced. Okay, now, so what you have to do is, you have to go on the left panel. Whenever there is a weekly assignments and everything is given, you have to go to the problem solving session, and you have to click on problem solving session recording. If you click on this particular thing, you will go redirected to a page which will contain the weekly course content. I mean, it will contain a Google Drive link which will have the particular week's problem solving sessions, I mean lecture PPT, and also it will contain the lecture video, the class video. It will there will be also the class video, so you can download the things from there. And if you have missed anything during the class due to poor network connection or anything else, you will find it from here. Okay, so today, as I have already mentioned, that it is our week 3 live session. So let us first see what are the contents of the week 3 for this course. So week 3 course contents are like this: hierarchical structure of proteins, secondary, tertiary and quaternary structure, Ramachandran plot and protein purification techniques. Protein purification techniques, we will discuss it in detail. Okay, and we have the introduction to enzymes and its kinetics, enzyme catalyzed reaction, and introduction to the catalyte activity of proteases. So these are the topics, so these are the things which we will discuss. So in our last class, what we have discussed, regarding the, basically we have discussed about the primary structure of the protein, how to identify what is a particular in a particular polypeptide chain, a primary structure of a protein, what are the amino acids which are linked together, what is the method for determination of the N-terminal amino acid, what is the thing to identify the C-terminal amino acids. Okay, then we have discussed about various enzymes which cut the protein chain in a, or the polypeptide chain in a particular spot, and how it works. All the things we have discussed in an earlier class. Also, we have discussed about the protection reprotection chemistry, and we have discussed a little bit about the oxidizing and reducing agents and so on. Okay, so today we will discuss the, particularly in that our last class was basically concentrated on the primary structure of the protein, right? So today what we will discuss, about the secondary, tertiary, quaternary structure of the protein, and the other topics related to those things. Okay, so let us start with our first question of today's session. So the first question of today's session, it says that which of the following statements is not true for beta bands? Your options are: number A, hydrogen bond stabilizes the beta band structure; number B, proline and glycine are frequently found in the beta turns; number C, it occurs at the protein surfaces; and number D, it gives a protein linearity rather than globularity. Which of the following statements is not true for the beta bands? Again, I will repeat the options. Number A, hydrogen bond stabilizes the beta band structure. Number B, proline and glycine are frequently found in the beta turns. Number C, it occurs at the protein surfaces. Number D, it gives protein linearity rather than globularity. Which of the following statements is not true for the beta bands? I will wait for 2 minutes, then I will check the chat box. Whatever you think is the correct answer, you can write in the chat box A, B, C, or D. After checking the answer, I will let you know what is the correct answer and we will discuss a little bit about it. Which of the following statements is not true regarding beta bands? Number A, hydrogen bond stabilizes the beta band structure. Number B, proline and glycine are frequently found in beta turns. Number C, it occurs at protein surfaces. Number D, it gives protein linearity rather than globularity. Which of the following statement is not true for beta bands? Let me check the chat box. So the question, answering the question during the class, so there is no marks regarding it. So that is why there is no problem. Okay. Okay, number D is the given answer. Let me check. So as I was saying that for answering the class, there is no marks. So there is no problem that even if your answer is wrong, there will be no negative marking. But the assignments and everything, these are very important. So you have to you have to answer the assignments very carefully because these are all marked assignments. Okay, the whatever is posted on the SWAM portal. So the correct answer for problem number 1 will be yes, it is number D. It gives the protein linearity rather than globularity. So beta bands permits the change of the direction of the peptide chain to get a folded structure. It gives the protein globularity rather than linearity. Hydrogen bond stabilizes the beta band structure. Proline and glycine are frequently found in the beta turns. Beta turns often promote the formation of the antiparallel beta sheets. They occur at the protein surface and involve four successive amino acid residues. So basically, as you can see, this is an example of beta band which contains successively four amino acid residues. Okay, this gives you the protein globularity rather than linearity. Protein and glycine are frequently found in these beta bands or beta turns. Okay, so this is the example number 1, which is the type 1 of the beta band, or this is the type 2 of the beta band. What is the difference between type 1 and type 2? As you can see here, this particular the group, the carbonyl group is turned in this side, and here it is on the outer side of this hydrogen bonding. Okay, so beta band, so basically it is one of the secondary structure of the proteins. So whenever we have a polypeptide chain, which is the primary structure of the protein, the amino acids are linked together with the amine bonds, right? So and then what happens, we get the polypeptide chain. These polypeptide chains can interact with each other to give us the secondary structure of a protein. The most important secondary structures are the alpha helix and the beta pleated sheets. We have discussed this thing already in our last class, if you remember. In the last class, the last question was related to the secondary structure of the protein, where I have discussed about the alpha helix and beta pleated sheet. So basically, this is the thing is that all the polypeptide chains, they interact with each other via hydrogen bonding to give either a alpha helix or a beta sheet structure. Okay, but there is another structure which is known as the beta band structure, which is I mean the less frequent, but it is there. Okay, so the important top things which you have to remember regarding the beta bands, it gives it gives the protein globularity rather than linearity. Hydrogen bonds are there which stabilizes the structure. Proline and glycine, they are frequently found, and also the beta turns often promote the formation of antiparallel beta sheet. It often occur on the protein surfaces and involve four successive amino acid residues. Okay, so these things regarding beta band you have to remember. So I hope there is no problem. If there is any doubt, you can let me know. Okay, so this was our first question. Now I will go to the next question. So the next question, let me just show you. If there is any doubt, any query, please let me know through the chat box. Okay, I am checking the chat box every now and then. So if you have any doubt or any query, you can let me know through the chat box. Now, problem number 2, it says that which amino acid is incorporated into the polypeptide chain to induce flexibility, and which amino acid is termed as the helix breaker. Your options are number A, alanine and glycine respectively, number B, glycine and proline, number C, proline and glycine, number D, histidine and proline. So, which amino acid is incorporated into the polypeptide chain to induce flexibility? All the four options, the first amino acids are the options for the the particular amino acid for to induce flexibility, and the second one is for the helix breaker, the amino acid. So you have to now identify which amino acid is gives the polypeptide chain flexibility and which amino acid acts as a helix breaker. Again, I will wait for 2 minutes. Read the question very carefully, read the options very carefully, and let me know A, B, C, or D which of the following you think is the correct answer for problem number 2. Which amino acid is incorporated into the polypeptide chain to induce flexibility, and which amino acid is termed as the helix breaker? Let me know in the chat box A, B, C, or D which of the following you think is the correct answer. Let me check if there are any answers. Okay, so I have received several answers. I think, okay, I have received only two answers. One is, both are saying that it is number B. Okay, let me check if number B is the correct answers. Others also, please try to answer the questions, because there is no negative marking, right? So there is no harm in answering the questions. Okay, so I have received only one option as answer, which is saying that number B, which says glycine is the amino acid which gives the polypeptide chain flexibility, and proline is termed as a helix breaker. So now let me check if it is the correct answer or not. Yes, it is correct answer. Glycine is the amino acid which is incorporated to induce flexibility, and proline is termed as a helix breaker. So this is the alpha helix structure, as I have already mentioned earlier also, that alpha helix is one of the second very important or crucial, one of the very crucial secondary structure of the protein. Okay, the polypeptide chain, when they interact with hydrogen bonding with each other, they form alpha helix and beta pleated sheet. So this is the structure of the alpha helix. Okay, as you can see, the distance between two turns of the alpha helix is 5.4 Armstrong, which contains 3.6 amino acid residues, and it is the top view of the from the, if you look at from the this angle, you will find it looks like this. Okay, and there is a hydrogen bonding in this like this, and the side chain residues, they actually move outward with the helix like this on the right angle. They face outward to the axis of the helix, so that there is no I mean the steric hindrance. So this is the alpha helix. So all the amino acid residues, they give the alpha helix stability in, but not the proline. Proline is termed as a helix breaker. Why? This is the structure of the proline. As you all know, the structure of proline is the different from our other amino acid. As you know, the proline structure is like this. So it is we have, there is no NH2 bond. We have only, so this is COOH. Okay, so this is the structure of proline, as you know. Okay, and you also know that proline does not have any NH2. Okay, so one hydrogen only is there on nitrogen. Once it forms a peptide bond or amide bond, so there is no hydrogen left for further hydrogen bond formation, like it happens in the other amino acids. As you can see, one hydrogen for the amine, it is it forms the peptide bond, and other hydrogen is there for the hydrogen bond formation, but it does not happen in case of the proline, because proline nitrogen, it is already secondary nitrogen, and it does not have any hydrogen after the peptide bond formation. That is why this particular proline, it cannot give the alpha helix stability. Okay, this is the reason why proline is known as the helix breaker. And glycine, as you know, the structure of glycine does not have any side chain residue. It is the only amino acid which has only two hydrogens on the alpha carbon, does not have any other residue. That is why glycine is a very flexible amino acid which can incorporate into the polypeptide chain to induce further flexibility. Okay. So proline and glycine are exceptional amino acids. Glycine does not have any substituent, so it is much more flexible. Whenever you want to bring flexibility in a polypeptide chain, you incorporate glycine. You will see that the flexible regions, there are glycines. And if you want to bring the rigidity, means if you want to disrupt this kind of system which is stabilized by the intramolecular hydrogen bond, then incorporate proline, since proline cannot participate in hydrogen bonding. So proline is basically also called the helix breaker. It breaks the helix. So if there is some proline going on in the helical shape and suddenly change the another shape, that all changeable point, proline must be present, because proline is one which does not allow any helix formation. Now apart from this alpha and beta sheet structure, so that we have already discussed earlier. So remember, glycine is the amino acid which gives the polypeptide chain or the much flexibility, and whenever there is a break in the alpha helix, you have to understand that there must be a proline residue present in that particular point. So I hope this problem was so you have understood the problem number 2. If there is any doubt, please let me know. Otherwise, I will move toward the next problem. Okay, now let me move forward. And so problem number 3, it says, which of the following statements is false regarding the determination of 3D structure of proteins? Your options are: number A, best way is to crystallize the protein. Number B, NMR can also provide a static conformation of the proteins. Number C, cryo-electron microscope can directly show the image, no need for crystallization. Number D, for NMR, highly concentrated solutions are needed. Which of the following statements is false regarding the determination of the 3D structure of proteins? Read the options very carefully. Whenever there is a true false type of question, the students, most of the students, they get confused. But read the options very carefully. I know you can answer the question correctly. Okay. Again, I will repeat, which of the following statements is false regarding the determination of 3D structures of protein. Okay. Number A, best way is to crystallize the protein. Number B, NMR can also provide the static conformation of proteins. Number C, cryo-electron microscope can directly show the image, no need for the crystallization. Number D, for NMR, highly concentrated solutions are needed. I will wait for 2 minutes again and then I will check the chat box. Which of the following statements is false for the determination of 3D structures of the protein? Let me know A, B, C or D. Which of the following you think is the correct answer? I will wait for a minute, then I will check the chat box. Let me check if there are any answer for this problem number 3. Okay, I received only one answer which is saying it is number D. Let me now check if number D is the correct answer or not. Others also please try to answer. Okay, so let us see what is the correct answer for problem number 3. The correct answer is it is actually number B, the statement which is false regarding the determination of 3D structure of proteins, which says that NMR can also provide the static conformation of proteins. Actually, NMR can provide the dynamic conformation of the protein. So, the best way to crystallize a protein is the best way for the determination of the 3D structure of protein. Okay, most of the times you will find that the protein crystal structures are there reported. So, there is a place which is known as the protein data bank. Okay, you can go to Google and search. So you will find the protein data bank which has the crystallized structures of protein. These are reported. You can easily download the crystal structure or you can see or visualize everything. Okay, and the cryo-electron microscope can also directly show the image without crystallizing also. And for NMR is also can give us the dynamic structure or conformation of protein, but for that highly concentrated solutions are required. Okay. So regarding the determination of 3D structure of protein, there are different techniques to determine it. One of the best way is the X-ray crystallography. If you can isolate a crystal structure of the protein, then it is the ultimate structural evidence. Methods of determination to complete 3D structure. Usually there are three types. One is the X-ray crystallography and it is the most reliable one. Then we have the NMR spectroscopy. Very high field NMR machine is needed. Through NMR spectroscopy, you can determine the 3D structure of the protein. However, there are some disadvantages. You cannot handle very large molecules in NMR, but at the same time, there are some advantages as well. In X-ray crystallography, if you cannot crystallize a protein, you cannot have the crystal structure. X-ray crystallography need crystals and the structure is a static one. So, we get the average structure in the crystal. But NMR is recorded in solution, so you can see exactly the dynamics of the system. But you need highly concentrated solution to get the signals. The peaks are actually very complicated ones. You should have isotopically labeled carbon, hydrogen, nitrogens and there are techniques to do that. By NMR you can do the structure analysis but not for very large proteins. Okay, up to 60 kilodalton you can do the structural elucidation. So why 3D structure of proteins are required, or why you have to determine the 3D structure of protein? Because these 3D structures of proteins are a precious source of the information, like the shape and domain structure, protein classification, prediction of the function of uncharacterized proteins, interactions with the other macromolecules, interactions with the small ligands, metal ions, nucleotides, substrate cofactors and inhibitors, evidence for enzyme mechanism, structure-based drug development, post-translational modification, disulfide bond in glycosylation, experimental evidence for the transmembrane domain. So these are the precious sources of information that can be derived from the 3D structure of the protein. Okay. So the correct answer was number B, which says that NMR also provide the static conformation of the proteins. No, the extracrystallography, it provides the static information, but the NMR gives the dynamics. Okay. And as I have mentioned, the protein data bank is the place where you can find the crystal structures of the protein. These are reported officially. Okay. Now let us go to the next problem. Problem number 4 says, which of the following protein purification process is not based on the charge? Your options are number A SDS page, number B ion exchange chromatography, number C native gel electrophoresis, number D isoelectric focusing. Which of the following protein purification process is not based on the charge? SDS page, ion exchange chromatography, native gel electrophoresis or isoelectric focusing? Again, I will give you 2 to 3 minutes and then I will check the chat box. Which one of the following protein purification process is not based on the charge? Number A SDS page, number B ion exchange chromatography, number C native gel electrophoresis, and number D isoelectric focusing. Which one of the following protein purification process is not based on the charge? SDS page, ion exchange chromatography, native gel electrophoresis or isoelectric focusing? Let me check the chat box. Okay, I have got two answers. One is saying number A, other is saying number C. Okay, let me check which of the following is the correct answer, number A or C, or there is any other answer. So the correct answer for problem number 4 is, yes, it is number A, SDS page. So SDS page or sodium dodecyl sulfate polyacrylamide gel electrophoresis is widely used technique for the separating proteins based on their molecular weight. Okay, so this is the thing that SDS page, it actually separates the particular protein based on their molecular weight, not the charge. Okay, other the techniques which are mentioned in the other options like the ion exchange chromatography, native gel electrophoresis, isoelectric focusing, all of these techniques they actually use the charge, but not the SDS page electrophoresis. SDS page technique, it uses the molecular weight. It involves the denaturing of the protein with SDS, which coats them with a negative charge, allowing the separation based on their size as they migrate through the polyacrylamide gel under an electric field. So proteins may or may not have overall charge. Some proteins have overall charge, some charge may be there, may not be there, but there is no consistent charge or charge by size relationship like we need. So we add sodium dodecyl sulfate. As you can see, the structure, it is very complicated one, but it has a negative end, and it is a and sodium there is present. So SGS has a negative charge overall. It binds with the protein evenly and keeps it linear. Okay, then there is a direct charge by length relationship. Any natural charge it has will get overpowered by the charge coating it gets from the SDS. Okay, then we can use the charge to send through the polyacrylamide gel mesh. As you can see, there is a gel mesh like this. There is a negative end here, there is a positive end, but the longer the chain get tangles up in the mesh and travel more slowly. And when you turn off the power, you haven't and when you have turn off the power, they haven't moved as far. To bigger the chain, they will they will stay here at the this side, and to the negative end of this particular gel. And because they are because of the bigger size, they cannot move through the mesh very well. But the smaller proteins, they get easily, they can move, and they will, you will find there is, they are at the lower end of the mesh. Okay, so this is the technique of the SDS page, the coating, and then the separation, and all these are based on the molecular weight. So here are more detailed explanation. Denaturing SDS is a detergent that unfolds the protein into the linear chain and coats them with the negative charge. Polyacrylamide gel, the gel acts as a molecular sieve, separating proteins based on their size and shape. Smaller the proteins migrate faster through the gel. Electrophoresis and electric field is applied, causing negatively charged proteins to migrate toward the positive electron. Okay, separation, the rate of the migration inversely proportional to the molecular weight of the proteins. Smaller the proteins move faster and travel faster further through the gel. Visualization, after electrophoresis, proteins are visualized using stains like pumice blue or silver stain, revealing the distinct protein bands at different location of the gel, as you can see like this. Okay. And key applications of the SDS page: protein purity assessment. SDS page can be used to determine if a protein sample is pure or contains other contaminating proteins. Protein molecular weight determination, by comparing the migration distance of the protein to a known protein standard, its approximate molecular weight can be estimated. Protein identification, SDS page can be used in the conjugation with other techniques like mass spectrometry to identify proteins. Protein analysis in food science, SDS page is used to analyze proteins in various food products such as cereal, dairy, and meat. Okay, so this is the very important technique, the SDS page. And question can be asked like to say what is the what is the theory behind it, what are the application, why SDS are used. This type of question can be asked if detailed question in case of the subjective question answer. Okay. So is there any doubt, or should I repeat, or should I move forward? If there is any doubt, please let me know. I will repeat. Okay. Then I will move to the next problem. So problem number 5, it says that which of the following statements is false for the gel filtration technique? Your options are: number A, it is also called the size exclusion chromatography. Number B, chemical polymers can be separated by using this technique. Number C, small size proteins will elute first, followed by larger size one. And number D, different sized cutoff gels are used to separate the proteins. Which of the following statements is false for gel filtration technique? Number A, it is also called the size exclusion chromatography. Number B, chemical polymers can also be separated by this technique. Number C, small size proteins will elute first, followed by the large size ones. Number D, different sized cutoff gels are used to separate the proteins. Read the options very carefully and then let me know in the chat box A, B, C or D which of the following you think is false regarding gel filtration technique. I will wait for a minute or two, then I will check the chat box. Which of the following statements is false regarding gel filtration technique? It is also called the size exclusion chromatography. Number A. Number B. Chemical polymers can also be separated by using this technique. Number C. Small size proteins will elute first, followed by the large size ones. Number D. Different sized cutoff gels are used to separate the proteins. Let me check the chat box. So for what are the answers? So I didn't receive any answer for this question. Okay. Let me show you the answer and let us discuss. So the statement which is false for gel filtration technique is it is number C, which says that small size proteins will elute first, followed by the large size ones. So what is a gel filtration chromatography? So chromatography encompasses a variety of techniques that involve a mobile phase flowing through the bed of the stationary phase. A mixture of solutes is resolved and separated as a result of such differences in affinity of the solutions for the mobile phase relative to the stationary phase. A solute that has more affinity for the mobile phase moves forward with the mobile phase, whereas a solute with more affinity for the stationary phase is retarded. Thus, solutes are separated into the bands based on the distribution between the phases. Molecules of biological interest are separated or resolved by the variety of chromatography techniques, including absorption chromatography, ion exchange chromatography, partition chromatography, paper or thin layer, gas liquid chromatography, molecular exclusion chromatography, which is also known as the molecular sieve or gel filtration. Okay, so gel filtration chromatography is also known as the molecular exclusion chromatography or molecular sieve chromatography also it can be termed as. Chromatography involves a mobile phase, a liquid or gas flowing through a stationary phase, which is can be solid or liquid. As you can see in the figure 1, this is the stationary phase. This is marked like this. These are the stationary phase, and the mobile phase is termed as M. Okay, and the solute flow is like this from top to bottom. Okay, the mixture of solutes are separated based on their solute components, degree of affinity to the mobile phase and the stationary phase. Now, if component 1 has a stronger affinity to the stationary phase than component 2, then component 2 will migrate to the column rapidly than component 1. Okay, so whenever which if let's say compound number A, it has a strong binding with the stationary phase than the compound number 2, then it is normal that compound 2 it will be eluted first, then after the only the compound 1 will elute it because it is more strongly bound to the stationary phase. So what happens in the size exclusion chromatography? So the stationary phase it actually has the pores like this, okay, and small molecules are able to enter the pores very easily, but the large molecule they are unable to enter the pore. So what happens? So whenever there is a mixture of a large or small molecules, the small molecules it will enter the pores and they will stay inside the pore and they will not, but the large molecule they cannot enter the pore, so they will move fast through the column and they will be eluted first, then the small size. So as you can see, the retention time is or the time which the particular component spends through the column is known as the retention time. Okay, so whenever so large compound they have less retention time because they cannot enter the pores of the pores of this stationary phase, so they will be eluted first, then the small compound which tightly bonds with the stationary phase, so it has the higher retention time. So the statement which was given like so let us see now, check all the statements. So number A, it is also called the size exclusion chromatography, which is correct. Chemical polymers can be also separated, it is also correct. Small proteins will elute first, followed by the large one, it is not correct because large proteins will be eluted first, then the small one. Different size cutoff gels are used to separate the protein, it is also true. Okay, so this was the correct answer for problem number 5. So I hope it is fine with all of you. Have do you have any doubt? If there is any doubt, you can let me know. Otherwise, I will move forward. Okay. Let me go to the next problem. Problem number 6, it says that in ion exchange chromatography, if the column is negatively charged, then which of the following statements is true? Your options are: number A, proteins with Pi less than pH will buffer of the buffer will elute. Number B, proteins with Pi greater than pH of the buffer will elute. Number C, proteins with Pi equal to pH of the buffer will elute. Number D, none of the above. So in ion exchange chromatography, if the column is negatively charged, then which of the following statement is true? Number A, proteins with Pi less than pH of the buffer will elute. Number B, proteins with Pi greater than the pH of the buffer will elute. Number C, proteins with Pi equal to pH of the buffer will elute. Number D, none of those, none of the above. Try to answer the question. I will wait for 2 minutes, then I will check the chat box. In ion exchange chromatography, if the column is negatively charged, then which of the following statement is true? Number A, proteins with Pi less than pH of buffer will elute. Number B, protein with Pi greater than pH of the buffer will elute. Number C, proteins with Pi equal to pH of the buffer will elute. Number D, none of them. Let me check the chat box now. Okay, so I have received two answers. One is number A, another is number B. Okay, let me check number A or number B. Two options I have received. Let me see which of the following is the correct answer. So, the correct answer will be, it is number A, proteins with Pi less than pH of buffer will elute. So purification of the protein by ion exchange chromatography. So if the column is negatively charged, for example carboxymethyl, then proteins with Pi less than pH of the buffer will be negatively charged and they will not bind to the column but elute. And proteins with the Pi is greater than pH of the buffer, it will be positively charged and it will not it will bind to the column. Okay, so this pH of the buffer and Pi, how the proteins behave with a particular pH of the buffer, we have already discussed in our very first class. class. Okay, so you have to remember these things. So proteins or amino acid that has the pi less than pH of the buffer, they will be negatively charged, and the proteins with the pi's greater than pH of the buffer, they will be positively charged. So these two things you have to remember. Okay, pi less than pH of the buffer will be negatively charged, and pi greater than pH of the buffer will be positively charged. So if the column is negatively charged and the proteins are also negatively charged, then the proteins will not bind and they will elute first. And if the opposite charge are there, like the column is negatively charged but the protein is positively charged, then it will bind to the column and they will not elute. So this is the principle of the ion exchange chromatography. So remembering the principles of each and every chromatographic technique, be it SDS page, be it ion exchange chromatography or gel exclusion, gel filtration chromatography. So remembering the particular principle is very, very important. Okay, so this is the example. So let's say the column or the particular column is negatively charged. So if the protein is positively charged, they will be immobilized on the negative surface, but the positively charged protein will be immobilized on the anionic surface. Okay, and eluted with a low salt gradient, and with the low the negatively charged proteins, they will be eluted with a low salt gradient, and the positively charged one, it will be eluted with the high salt gradient. Okay, so these are the example of the ion exchange chromatography. Now let me go to the next problem. Problem number 7, it says that which of the following statements is true about the two-dimensional electrophoresis? Your options are: number A, proteins are separated based on their stationary and mobile phase. Number B, proteins are separated based on both molecular weight and pi. Number C, proteins are separated based on size of the eluate. Number D, proteins are separated based on the charge of the ion. Which of the following statements is true about the two-dimensional electrophoresis? Again, I will repeat the options. Which of the following statements is true about the two-dimensional electrophoresis? Your options are: number A, proteins are separated based on the stationary and the mobile phase. Number B, proteins are separated based on the both molecular weight and the pi. Number C, proteins are separated based on the size of the eluent. And number D, proteins are separated based on the charge of the ions. Let me know A, B, C or D, which of the following statement you think is true regarding the two-dimensional electrophoresis. So all these are actually the separation techniques or the techniques by which the proteins are separated. Okay, and each and every technique has a particular principle. So when in the case of the whenever there is a these are MCQs are intended to for you for me to make you able to answer your assignments, and also I am discussing all the things in detail so that in final examination, if you are asked the subjective questions related to these things, you can easily answer. So remembering the principle of all the techniques are very important. What is the principle of the techniques? Okay, separation techniques, how it is, it happens, and everything with figures is also important to for you to understand and to remember. Let me know which of the following you think is the correct answer for problem number 7. I will wait for a minute, then I will check the chat box. Okay. Let me check the chat box. I think there will be answers. Okay, so I have got one answer which is saying it is number B. Let me check if number B is the correct answer or not, and then let me discuss about it. So In the two-dimensional electrophoresis, the statement which is true regarding it will be very good. It is number B. Proteins are separated based on their molecular weight and the PI. PI means the isoelectric point, right? So, what is the overview of two-dimensional electrophoresis? So, basically, in 2D gel electrophoresis, proteins are separated as per their isoelectric point and also the protein mass. Okay, separation of the proteins by isoelectric point is also called the isoelectric focusing. This is very important term. What is isoelectric focusing? You have to remember it. Okay. So separation of the proteins based on their pH, based on their isoelectric point is known as isoelectric focusing or IEF. Okay, when the gradient of pH is applied to a gel and an electric potential is applied across the gel, making one more positive than the other, at all pH values other than their isoelectric point, proteins will be charged, right? We all know that at at the isoelectric point, the protein is neutral, but at the all pH values other than their isoelectric point, the proteins will be charged. If they are positively charged, they will be pulled toward the negative end of the gel, and if they are the negatively charged, they will be pulled to the positive end of the gel. The proteins applied in the first dimension will move along the gel and will accumulate at the isoelectric point. At this the point will we have overall charge of the protein, the zero as the neutral. Separating the proteins by the mass in the gel treated with SDS along with the page. Okay, the SDS page that it denatures the protein and then unfolds it into the long and straight molecules and binds to the number of SDS is roughly proportional to the protein's length. Because a protein's length when unfolded is roughly proportional to the mass, since the SDS molecules are negatively charged, the result will all the proteins will have approximately same mass to charge ratio. So the first one is the isoelectric focusing. As you can see, the bands are created based on the isoelectric point of the particular protein. Then this particular thing is placed in a SDS gel polyacrylamide, and it is then the second dimension. What happened? It is separated based on their charge. Okay, so this is at this from left to right. Okay, it goes like decreasing the PI, and from top to bottom it goes like the decreasing the molecular weight. So that's how the 2D electrophoresis happens. So proteins are separated based on their both molecular weight and PI in case of our 2D gel electrophoresis. Okay. So is it clear or there is any issue? Till now, is there any issue at any question? You can let me know. Otherwise, I will move forward. Okay, so I hope there is no problem. Then let me go to the next question. So problem number 8, it says that which of the following is not a weak interaction? Your options are number A, pi-pi stacking, number B, hydrogen bonding interaction, number C, salt bridge interaction, number D, disulfide bond. Which of the following is not a weak interaction? Pi-pi stacking, hydrogen bonding interaction, salt bridge interaction, or disulfide bond? Let me know in the chat box A, B, C, or D which of the following you think is the correct answer. Which of the following is not a weak interaction? Number A, pi-pi stacking, number B, hydrogen bonding interaction, number C, salt bridge interaction, number D, disulfide bond. Which of the following is not a weak interaction? Let me check if there are any answers in the chat box. Okay, no answer for this problem number 8. Okay, let me discuss and let me show you the correct answer and let us discuss about it. So the following interaction which is not a weak interaction is, it is a disulfide bond. Okay, so this is the pi-pi stacking interaction. Let me just first give you all the things. Now let us discuss. So pi-pi stacking interaction. So basically, it knows not any I mean covalent bond formation. So basically, it is an interaction between the pi electron clouds of two benzene ring or two aromatic systems which are on top to each other. Okay. Then hydrogen bond, it is also a kind of the it is not any kind of the proper bond. Okay, it is only a electrostatic interaction between a between a hydrogen and also between the hydrogen bond donor and acceptor. Electrostatic salt bridge interactions is also the electrostatic interaction between a positive and negative charge. So all these hydrogen bonding interaction, pi-pi stacking interaction, and electrostatic salt bridge interactions are not a particular bond, but rather they are the basically the interaction between two opposite charges or sometimes the electron clouds. Okay, but in case of the disulfide bond, what happens? It is a proper covalent bond. Okay, it is a sulfur sulfur bond which happens between two cysteine. When two cysteines are in close proximity, it will form a close bond. Okay, and this disulfide bond is also known as the disulfide bond or dicysteine bond, and it significantly stabilizes the tertiary structure of protein. Okay, and it is a covalent bond. Okay, so we have a cysteine residue, we have the cysteine residue have the SH. We all know SH are present in the cysteine residue. So what will happen? This is they will form this kind of sulfur-sulfur bond, which is very stable in nature and which is very strong in character. So these are the so it is not a kind of normal weak interaction. Okay, other interaction which I mentioned like pi-pi stacking, electrostatic, and hydrogen bonding. These are kind of mere interaction, not a proper bond. But disulfide bonds are strong bonds. These are that are formed. Okay. So this is the correct answer is number D, disulfide bond, which is not a weak interaction. So I hope it is clear. As I am saying repeatedly, if there is any doubt, please let me know, and also please try to answer the questions. Okay. Otherwise, I won't understand if you are following the class or not. Let me go to the next problem then. If there is any no doubt. Problem number 9, it says which one of the following statements regarding the enzyme is true? Your options are number A, enzymes are one kind of proteins. Number B, they cannot change the rate of the reaction. Number C, they can get consumed in the course of the reaction. Number D, they are not site specific in nature. Which of the following statements regarding enzyme is true? Again, I will repeat. Number A, enzymes are one kind of proteins. Number B, they cannot change the rate of the reaction. Number C, they get consumed into the course of the reaction. Number D, they are not size specific in nature. Which of the following statements is regarding the enzyme is true? Let me know in the chat box A, B, C or D, which of the following you think is the true regarding the case of the enzymes. Which of the following statement regarding enzyme is true? Number A, enzymes are one kind of proteins. Number B, they cannot change the rate of the reaction. Number C, they get consumed in the course of the reaction. Number D, they are not site specific in nature. Let me check the chat box. Okay, I got one answer which is saying it is number A. So the statement which is true regarding enzyme is yes, it is number A. Enzymes are one kind of proteins. So the process of enzyme catalysis, this figure actually summarizes the process of enzyme catalysis. So here we have our enzyme which has a particular site where our substrate is binding, and after the binding we have the reaction which forms the product and then the release. So binding, reaction, release, these are the three steps of an enzyme catalyzed reaction and the process of the enzyme catalysis. So basically enzymes provide a reaction surface and a suitable environment for the reaction. They bring the reactants together, position them correctly so that they can easily attain their transition state configurations. Enzymes weaken the bonds of the reactants and they may participate in the reaction mechanism. Enzymes form strong interaction with the transition state with the substrate or the product. So this is the graphs demonstrating the stabilization over the reaction transition state by the enzyme. So this is a normal transition, normal graph for normal transition state of a particular enzyme catalyzed reaction. So this is the starting material, this is the product, this is the delta G, which is the amount of stabilization from the starting material to the product, and this is the activation energy, the energy which the starting materials they have to attain to go to the transition state for the product formation. So what the and the y-axis, it defines the free energy and the x-axis is the reaction progress. With enzyme, what happens? There is a new transition state that is formed. So the enzyme, the starting materials now they need less activation energy to cross the transition state barrier to form the product. So that what the enzyme does. Okay. So basically the enzymes, so the let us now look at the statement. So enzymes are kind of protein, it is true, all the enzymes are one kind of protein. So they cannot change the rate of reaction, it is false. Basically enzymes, they alter the rate of the reaction, they get consumed in the course, it is also false. They do not get consumed, they remain intact at the end of the reaction. They are not site specific in nature, it is also not true. Enzymes are quite site specific in nature. So there are two theories. One is called as the lock and key theory, where the substrate almost it fits perfectly with the enzyme's active site. And induced fit theory, where the substrate does not fit perfectly, but they induce the enzymes so that it can it fits properly to the active site. So these are the two hypotheses for the enzyme substrate enzyme binding, which we will discuss in our upcoming slides. Okay. So yeah, number A was the correct answer for problem number 9. Now let us go to the next problem. Problem number 10 says which statement is false regarding the induced fit hypothesis. Your options are number A, substrate specificity is range is high. Number B, the active site is rigid. Number C, more accepted model for the enzyme substrate complex than lock and key. And number D, conformation change occurs in enzyme and substrate. Which statements is false regarding induced fit hypothesis? Number A, substrate specificity range is high. Number B, active site is rigid. Number C, more accepted model for enzyme substrate complex than lock and key. Number D, conformational change occurs in the enzyme and the substrate. Which statement is false regarding the induced fit hypothesis? Number A, substrate specificity range is high. Number B, active site is rigid. Number C, more accepted model for enzyme substrate complex than lock and key. Number D, conformational change occurs in enzyme and substrate. Let me check the chat box. Okay, so I have received one answer which is saying it is number C. Let me check if number C is the correct answer or not. So now let us, what is this? Yeah. So the correct answer, the statement which is false regarding induced fit hypothesis is, it is number B, which says that the active site is rigid. Okay, so actually in case of the induced fit hypothesis, the site specificity range is high, but the active site, it is not rigid. As I have mentioned in the earlier question also, that in case of lock and key theory, which we suppose that the active site is rigid and the substrate, it fits perfectly with the active site like a lock and a key. Right, but in case of the induced fit hypothesis, we have actually it is actually thought that the substrate enzyme substrate enzymes I mean active site is not rigid. It is quite flexible, so that and it actually makes it so that the substrate can accommodate to this particular space. Okay. It is more accepted model than lock and key theory. Okay, and the conformation change occurs in both the substrate and enzyme. So induced fit in presence of substrate, the active site undergoes a change in its conformation to allow the better fit between the active site and substrate. This means the enzymes are highly specific for the reaction they catalyze. Okay, so this is an example, substrate entering the active site of enzyme. As you can see, the active site looks like this, but in presence of substrate, it changes its shape slightly as the substrate binds. Okay, and then enzyme the product complex formation happens, and the product is leaves the active site of enzyme, and the active site attains the previous conformation before the reaction. Okay, so this is the induced fit hypothesis, and actually it is more accepted than the lock and key theory. Okay, so this was the problem number 10. Now let me go to the next problem. Is there, if there is any is there is any doubt? Let me know if there is any doubt regarding the question number 10. Otherwise, I will move forward. Okay. Problem number 11, it says that which of the following statements is not true for Km and Vmax? Number A, Vmax is the maximum rate of that the particular enzyme catalyte reaction can proceed. Number B, a small value of Km tells us that the enzyme binds strongly to the substrate. Number C, Km is the concentration at which the rate of the reaction reaches the Vmax. Number D, large value of Km tells us that the enzyme shows little specificity for the given substrate. Again, I will repeat, identify the statement which is not true for Km and Vmax. Number A, Vmax is the maximum rate at which the particular enzyme catalyzed reaction can proceed. Number B, a small value of Km tells us that the enzyme binds strongly to its substrate. Number C, Km is the concentration of the substrate at which the rate of the reaction reaches the Vmax. Number D, a large value of Km tells us that an enzyme shows little specificity for a given substrate. Let me know in chat box A, B, C or D which of the following statement you think is not true regarding the Km and Vmax. Which of the following statements is not true regarding Km and Vmax? I will wait for a minute, then I will check the chat box. Number A, Vmax is the maximum rate at which a particular enzyme catalyzed reaction can proceed. Number B, small value of Km tells us that the enzyme binds strongly to its substrate. Number C, Km is the concentration of the substrate at which the rate of the reaction reaches Vmax. Number D, large value of Km tells us that an enzyme shows little specificity for the given substrate. Let me check the chat box. So, are there any answer for problem number 11? Okay, so I find one answer which is saying number D. Let me check if number D is the correct answer. So the statement which is not true regarding the Km and Vmax will be, it is number C. Okay, Km is the concentration of substrate at which the rate of reaction reaches Vmax. It is not true. Okay, let me just show you. So this is about the Michaelis-Menten curve. Okay, so it is the Michaelis-Menten curve for the which is generates from the Michaelis-Menten enzyme uh, kinetics. Okay, so, and this is the graph which actually represents the enzyme catalysis. So this on the y-axis we have the rate of the reaction, on the x-axis we have the substrate concentration. So basically Km is the concentration at which the particular reaction rate it actually have this half reaches to the half of the Vmax. Okay, so Km is the substrate concentration when the reaction reaches to the half of the Vmax or half of the maximum reaction rate. Okay, and this at the beginning of the reaction, as you can see, now this is the first order reaction, and then in between it we get the mixed order reaction, and at the end it gets the now at the higher, so at the lower substrate concentration the rate of the enzyme catalyzed reaction is of the first order. At the middle it has the mixed order reaction, and then at the higher substrate concentration the rate of the reaction reaches to the it does not it gets independence of the substrate concentration. As you can see, it gets to the zero order, because when V0 equal to Vmax there is no change of the rate by increasing the substrate concentration. But at the beginning when the substrate concentration is low, there is a steady change of the rate. So it gets so this is the equation which is notes as a V0 equal to Vmax into substrate concentration divided by Km. Okay, which is the for the at the beginning when the substrate concentration is low. So this is the Michaelis-Menten equation basically. Okay, so this equation is very important. You have to remember it. You have to understand this equation. V0 is the initial velocity mole per time. Vmax is the maximum velocity. It is also in mole per time. Okay, S is the substrate concentration in the molar, and Km is the substrate concentration at half Vmax. Okay, so at the beginning, whenever at this, when the substrate concentration at the initial stages when substrate concentration much less than the value of Km, so we can neglect the substrate concentration from the denominator and we get this equation. Okay, and when their substrate concentration is very high, okay, at the at this position, when the substrate concentration is very high, so we can neglect the Km, and if we neglect the Km, then substitute this S and S, they cancel out and we get this equation. Okay, so this remembering this equation is very important. This Michaelis-Menten equation, how it happens when the substrate concentration is low and how it looks when the substrate concentration is high. So these things which you have to remember regarding the Michaelis-Menten enzyme kinetics. Okay. Now let me go to the next problem. Problem number 12 says in Michaelis-Menten equation at low substrate concentration the rate of the reaction becomes number A second order with respect to the substrate concentration, number B zero order with respect to the substrate concentration, number C first order with respect to the substrate concentration, number D zero order with respect to the enzyme concentration. If you have paid attention to the last question, I know all of you can answer the problem number 12 correctly, because I have literally shown you how it happens. I will give you 1 minute, then I will check the chat box. Okay. In Michaelis-Menten equation at low substrate concentration, rate of the reaction becomes what? Number A, second order with respect to substrate concentration. Number B, zero order with respect to substrate concentration. Number C, first order with respect to the substrate concentration. Number D, zero order with respect to the enzyme concentration. In Michaelis-Menten equation at low substrate concentration, rate of the reaction becomes what? Let me check the chat box. Number C, I have got the answer. Let me check. Anyways, I have already discussed it in the earlier slides, so I know you can answer it correctly. So let me just remove these things. Okay. One more. So the correct answer will be yes, number C, first order with respect to the substrate concentration. So this is the Michaelis-Menten graph. Okay. This is the on the y-axis we have the velocity of reaction, on the x-axis we have the substrate concentration. I have mentioned when the substrate concentration is low, means this when lots of substrates are when lots of enzymes are present, then the reaction is follows the first order kinetics. Okay. Then there is a competition with the enzyme as the substrate concentration is increases, there is this competition between the enzyme molecules, and at the high substrate concentration we get the zero order because almost all enzymes are now get occupied with the substrate. Okay, so how to find, so let's say you do not remember this thing, so how to find it out? So the Michaelis-Menten equation we all know that V0 equal to your V max. I am writing it as a Vm only into the substrate concentration S divided by Km plus S. Right, this is our Michaelis-Menten equation. So when the substrate concentration is much less than Km, means at the low substrate concentration, what happens from this equation? You can ignore the Km, sorry, you can ignore the substrate concentration, then you will get this equation like this: V0 equal to V max by Km into the substrate concentration, because we are ignoring the substrate concentration in the denominator, which is the first order reaction. Okay, and when the substrate concentration is much greater than the Km, what happens? We can ignore Km from the denominator, then substrate concentration and if this S and S are cancelling out, then V0 equal to, we get the V max, which is a zero order reaction. Okay, so that's how you have to calculate these things. You have to find out this thing. This is very easy. You can remember it easily theoretically or practically. You can find it out from the Michaelis-Menten equation also. Okay, now let me go to the next problem. Problem number 13 says, what is the ratio of V max by V0 when the substrate concentration is equal to 9 km? Your options are number A 9 by 10, number B 10 by 9, number C 9 by 8, number D 1 by 9. What is the ratio of Vmax by V0 when substrate concentration equal to 9 km? I will give you 2 to 3 minutes for this question because it is a mathematical question. It is very easy. You just have to write the values in the Michaelis-Menten equation and you will get the answer. Take 2 minutes. Try to do it using your copy and pen. And then let me know in the chat box A, B, C, and D which of the following you think is the correct answer. This type of small mathematical questions can also be given to you, so you have to practice. What is the ratio of Vmax by V0 when substrate concentration equal to 9 km? What is the ratio of Vmax by V0 when S equal to 9 km? Let me check the chat box now. Okay, I got one answer which is saying number A. I don't know it is for which question. Yeah, it is for this question only. Okay, let me show how to do this type of problem, how to solve this type of problem. So basically the correct answer is number B, which says that it is 10 by 9. So this is the Michaelis-Menten equation. Okay, V0 equal to Vmax into substrate concentration divided by Km plus S. V0 is the initial velocity and S is the substrate concentration. Vmax equal to maximum velocity. Km is the substrate concentration at half Vmax. Okay, so Michaelis-Menten equation, if we rearrange this equation, we just take Vmax to this left hand side, we will find V0 by Vmax equal to substrate concentration S divided by Km plus S. According to question, S equal to 9 km. So we will just replace the S with 9 km in this equation. So we will find 9 km divided by km plus 9 km means 9 km divided by 10 km. Km cancel out and we get the 9 by 10. So V0 by Vmax equal to 9 by 10 and Vmax by V0 equal to 10 by 9. Okay. So that is how you have to solve this type of question. It is understandable? Is it fine, or do I need to repeat this? It's very easy. This type of questions are actually very easy. You only have to place all the things correctly, and then you will find the correct answer. Okay. So the correct answer was number B. Now problem number 14, it says in which family of enzyme does fumarase enzyme belong? Your options are number A ligase, number B lyase, number C isomerase, number D transferase. In which family of enzymes does fumarase enzyme belong? Ligase, lyase, isomerase, transferase. Let me know A, B, C or D. Which of the following you think is the correct answer? I will wait for some time. In which family of enzyme does fumarase enzyme belong? Ligase, lyase, isomerase, transferase. Let me check the chat box now. Okay, I got one answer which says it is number D. Let me check if number D is the correct answer or not. So the family of enzyme where fumarase enzyme belong is, it is number B, lyase. So fumarase, what does the fumarase do? Basically, fumarase transform the fumarate to S-malate. Okay. So what are the enzyme classes? So we have oxidoreductases, which catalyzes the oxidation reaction or the redox oxidation reaction is the redox reaction. Example is the lactate dehydrogenase. We have transferases. They move the chemical group like hexokinase. We have hydrolases like hydrolysis, bond cleavage with the transfer of functional group of water like lysozyme. Lyases, non-hydrolytic bond cleavage. Fumarase is the example of lyase which reacts, which catalyzes the non-hydrolytic bond cleavage. Okay, it's like this kind of. Now then we have the isomerase, the intermolecular group transfer or isomerization, like triose phosphate isomerase. Then we have ligases, the synthesis of new covalent bond between substrate using ATP hydrolysis, like RNA polymerase. So what is the difference between lyases and ligases? So basically lyases are the enzyme that catalyze bond breaking reaction. Ligases are the class of enzyme that involve in catalyzing bond formation reaction. Lyases cause the forming of double bond. Ligases cause the forming of chemical bonds. They involved in the elimination reaction. They are involved in the hydrolysis reaction. Lyases act on a single substrate per reaction. Ligases act on the two substrate or reactants per reaction. Lyases are useful in glycolysis. Ligases are useful, important in the DNA replication. Okay, so these are the comparison between the lyase and ligase. Now let us go to the next problem. Problem number 15 says, in which family of enzyme does hexokinase enzyme belong? Your options are number A ligase, number B hydrolase, number C isomerase, and number D transferase. In which family of enzyme does the hexokinase enzyme belong? Ligase, hydrolase, isomerase, or transferase? In which family of enzyme does hexokinase enzyme belong? Ligase, hydrolase, isomerase, transferase? Let me check the chat box. Okay, I got, there is no answer. Okay, let me check which of the following is the correct answer. So hexokinase belongs to transferase. Okay, transferase is the family of enzyme where the hexokinase belongs. Again, have already shown you this particular table in the earlier slide. I am not going to repeat this thing. Only let us discuss a little bit about hexokinase. So hexokinase is an enzyme which catalyzes the phosphorylation of glucose to glucose 6-phosphate. It is also a non-specific enzyme capable of catalyzing the phosphorylation reaction of D-xylose. With the kinetic data provided below, describe whether the hexokinase is a so this is this. Let us just skip it. So hexokinase basically it catalyzes the transformation of beta glucose, so beta glucose to glucose 6-phosphate. Okay, it catalyzes the phosphorylation. As you can see, hexokinase where there is this is one of the transferases which moves the chemical groups. Okay, so here it moves the phosphate group to from this to this. Classification and characterization of the transferases. Transferases are enzymes which catalyze the reaction of the transfer of the groups of the atoms from one substrate to another. Almost all transferases are two component enzyme. They are contained in the cells only on the depend on the character transported groups. There are some subclasses of transferase, amino, methyl, acyl, and phosphotransferases. Okay, so these are the details about the transferases. Now problem number 16. The Lineweaver-Bark plot is associated with number A, straight line with the slope of minus km by Vmax, number B, parabola with the intercept of 1 by Vmax, number C, straight line with the slope of km by Vmax, and number D, parabola with the intercept of minus 1 by Vmax. The Lineweaver-Bark plot is associated with which of the followings? Number A, straight line with a slope of minus km by Vmax. Number B, parabola with the intercept of 1 by Vmax. Number C, straight line with the slope of km by Vmax. Number D, parabola with the intercept of minus 1 by Vmax. Let me know in the chat box A, B, C or D which of the following you think is the correct answer. Lineweaver bark plot is associated with what? Number A, straight line with the slope of minus km by Vmax. Number B, parabola with the intercept of 1 by Vmax. Number C, straight line with the slope of km by Vmax. And number D, parabola with the intercept of minus 1 by Vmax. Lineweaver bark plot is associated with what? Let me check the chat box. Okay, we don't have any answer. Okay, let us discuss what is Lineweaver bark plot because it is very important. It is equally important as the Michaelis-Menten curve. Okay. So it is a straight line with a slope of km by Vmax. So this is the Lineweaver bark plot. So the problem associated with the Michaelis-Menten kinetics is the fact that there may not be sufficient data points to determine whether the curve of Michaelis-Menten plot has reached to the maximum value or not. This means that values for the maximum rate and km are likely to be inaccurate. More accurate values of these properties can be obtained by plotting reciprocals of the rate and the substrate concentration to give a Lineweaver bark plot which looks like this. The rate of the equation is 1 by rate equal to km by rate by rate max into substrate concentration plus 1 by V max. So rate means we can also say it as a V. Okay, so the maximum rate can be obtained from the intersect of y axis and km. Okay. So, how to reach to the Lineweaver bark plot from the Michaelis-Menten equation. So, Michaelis-Menten equation we have like this: V0 equal to V max into substrate concentration divided by Km plus substrate concentration. You only have to do is take reciprocal of both sides. So, what we will get? We will get 1 by V0 equal to Km by V max into the substrate concentration plus 1 by Vmax. Okay, if you rearrange this thing, you will find this, and if you compare it with y equal to mx plus c, then you will find that y is the 1 by v0, m is the slope which is km by vmax, and sc is the intercept which is the 1 by vmax. Okay, so that is Lineweaver bark plot, how to the determination from the Michaelis-Menten equation itself. Okay. Now let us go to the next problem. Problem number 17 says, according to Ramchandran plot, which region is statically disallowed for all amino acids except glycine? Number A, red region, number B, yellow region, number C, white region, number D, all of these. According to Ramchandran plot, which region is statically disallowed for all the amino acids except glycine? Red region, yellow region, white region, or all of these? I will again wait for 2 minutes, then I will check the chat box and show you what is the correct answer. According to Ramchandra plot, which region is statically disallowed for all amino acid except glycine? Let me check the chat box. Okay, I got one answer which is saying it is number B. Let me check if number B is the correct answer for this problem number 17 or not. So the correct answer, the region which is sterically disallowed for all amino acid except glycine will be, it is the number C, white region. So what is the Ramachandran plot? The Ramachandran plot is also known as the Ramachandran diagram or a Phi-Psi diagram, is also originally developed in the 1963 by G. N. Ramachandran. It looks like this. The white region, this is sterically disallowed for the all amino acid except the glycine, the white regions. Okay, the red region, this and this are all region allowed regions, namely A helical and B sheet conformation. Yellow regions are outer limit. Okay, so it also provides the overview of allowed and disallowed regions for the torsion angle values and serve as an important indicator of the quality of protein three-dimensional structure. Torsion angles are among the most important local structural parameters that control the protein folding. The third possible torsion angle within the protein backbone is also known as omega. So there are two torsion angles of the polypeptide chain described in the rotation of the polypeptide backbone around the bond, name N alpha. Okay, N alpha, which is known as phi, and C alpha C. N C alpha, and this is the one, N and C alpha, which is known as phi, and C alpha C, which is known as the psi. And there is another which is C carbonyl carbon and nitrogen, which is known as the omega. Significance of Ramachandran plot. Ramachandran plot was the first verification tool for the protein structures. It displays the dihedral angles phi and the psi of all residues. It is a very powerful tool to identify errors in protein structures. It has become a standard tool in determination of the polar protein structure and in the defining secondary structures in terms of the alpha helices and beta sheet contents. It allows the display of distribution of residues in the protein in terms of the phi and psi angles. The region in the plot helps to identifying residues that are allowed and disallowed regions in the Ramachandran plot. Okay. So this was all about the Ramachandran plot. Let us move to the next problem. Problem number 18 is a mathematical question. An enzyme has a Km value of 7 millimolar and the rate of the reaction is 150 millimole per minute when the substrate concentration is 0.5 millimolar. When the enzyme is saturated with the substrate, what is the maximum reaction rate it can achieve? Your options are number A 2500 millimole per minute, number B 1100 millimole per minute, number C 1250 millimole per minute, and number D 2250 millimole per minute. If the enzyme has a Km value of 7 millimolar and the rate of the reaction is 150 millimole per minute when the substrate concentration is 0.5 millimolar, then the reaction, when the reaction is saturated with the substrate, what is the maximum rate of the reaction? Try to solve this question. I will give you 2 minutes and then I will check the chat box. An enzyme has a Km value of 7 millimolar and the rate of reaction is 150 millimole per minute and the substrate concentration is 0.5 millimolar. Just you have to put all the values in the Michaelis-Menten equation and you will get the answer. What will be the maximum reaction rate it can achieve? Let me check the chat box. Okay, there is no answer. Okay, let me show you how to solve this kind of problem. So the correct answer will be number D, 2250 millimole per minute. This is Michaelis-Menten equation. I have shown you it couple of times. So according to the problem, Km is 7 millimolar, right? Substrate concentration is 0.5 millimolar when V0 equal to 150 millimole per minute. Now, hence you have to just put all the values like V0 equal to Vmax into substrate concentration divided by Km plus S. Okay, so Vmax it will be V0 into Km plus S divided by substrate concentration, which is 150 millimole per minute, which is the V0, and it is multiplied by 7 plus 0.5 millimolar divided by 0.5 millimolar, the complete thing, which will give you 2250 millimole per minute. Hence, the maximum reaction rate achieved by the enzyme is 2250. It is very easy. Well, you just have to put all the values to the Michaelis-Menten equation and you will get the answer. According to the question, V0 is the substrate reaction rate 150 millimole per minute at the substrate concentration is equal to 0.5 millimolar and Km is 7 millimolar. Only you have to find out Vmax. So this is how you have to solve this kind of question, and it is actually very easy. One or two times you will practice. When I will share you the PPT, I think you will find it very easy. Okay. Now let me go to the next problem. Problem number 19. In the TCA cycle, the conversion of the malate to oxaloacetate is catalyzed by dash. Fill in the blanks. Your options are number A, malate dehydrogenase, number B, malate dehalogenase, number C, oxaloacetate synthase, and number D, aconitase. In the TCA cycle, the conversion of malate to oxaloacetate is catalyzed by dash. Number A, malate dehydrogenase, number B, malate dehalogenase, number C, oxaloacetate synthase, number D, aconitase. In the TCA cycle, the conversion of malate to oxaloacetate is catalyzed by what? Malate dehydrogenase, malate dehalogenase, oxaloacetate synthase, or aconitase. Let me wait for a minute, then I will check the chat box and after that I will discuss it. So, in TCA cycle, what is the enzyme that catalyzes the malate to oxaloacetate conversion? Let me check the chat box. Okay, I got one answer which says it's number C. Let me check if number C is the correct answer or not. So, the correct answer will be number A, malate dehydrogenase. So, malate to oxaloacetate conversion is catalyzed by the malate dehydrogenase, okay, and the cofactor is NAD plus, which is actually converted to NADH and H plus. Okay, so it known as the dehydrogenase because it removes two hydrogens or it removes hydrogens from the structure of malate. Okay, and you can see here we have an alcohol and it is catalyzed to the ketone group. So this is the TCA cycle. Okay, so first pyruvate is converted to acetyl-CoA with the pyruvate dehydrogenase. Then this acetyl-CoA is incorporated to the citrate, which is converted to the oxaloacetate is converted to the citrate, and the acetyl-CoA is also incorporated here as a cofactor. And the enzyme is the citrate synthase. Citrate is converted to the isocitrate with the help of aconitase. Isocitrate to the oxalosuccinate with the isocitrate dehydrogenase, then oxalosuccinate to convert it to alpha ketoglutarate with the isocitrate dehydrogenase, Then alpha ketoglutarate to succinyl-CoA with the alpha ketoglutarase dehydrogenase, succinyl-CoA to succinate with the GTP succinyl-CoA synthetase, then succinate to the fumarate with the succinate dehydrogenase, fumarate to malate with the fumarase, malate to oxaloacetate with the malate dehydrogenase. So this is the TCA cycle and all the enzyme activities associated with it along with their cofactors. Okay. So remembering the enzyme and cofactors are also equally important whenever you are studying an enzyme catalyzed reaction. Remembering enzyme is important as well as remembering the cofactors are similarly very important. Okay. Now, last question of today's session, which says, which of the following spectroscopic techniques requires a deuterated solvent for the experiment? Your options are number A, NMR, number B, IR, number C, UV, number D, SCXRD. Which of the following spectroscopic techniques require the deuterated solvent for the experiment? NMR, IR, UV, or SCXRD? This is the last question of today's session. Let me know A, B, C or D which of the following will be the correct answer. Which of the following spectroscopic techniques require the deuterated solvent for the experiment? NMR, IR, UV or SCXRD? Which of the following spectroscopic techniques requires a deuterated solvent for the experiment? Let me check the chat box. Okay, I got one answer which is saying it is number A. Let me check if number A is the correct answer or not. So the technique which requires deuterated solvent for the experiment is, very good, it is number A, NMR. So NMR is a technique by which you can detect the chemically distinct hydrogen, carbons, and all other elements can be also be distinguished. Like, okay, if the compound is NMR active, so this technique requires the deuterated solvent. So okay, so why deuterated solvents is required? So first, let us see what is the list of the solvents. So like CDCl3, it is the from non-polar to polar compound. This solvent can dissolve many different types of organic molecule. However, it is not great if dissolving the salt. If the sample recovery is interest, then the solvent is great the choice because it can be easily removed. TMS or D6 can dissolve a large range of compounds and relatively higher heat can be applied to dissolve the compound. Though this solvent is relatively viscous and can be oxidizing agent to some compound, also value of the solvent can have strong affinity to water. D2O, polar solvent will exchange with the labile proton and the solvent is ideal for dissolving salts and sugars. A drop of the solvent is like in others like DMS or D6 and CDCl3 can remove the labile protons from the spectra. CD3OD, which is deuterated methanol, a polar solvent with a high affinity for water, pairs well with salt and exchange with the labile protons, displays a water peak and residual peak in proton NMR. Due to polarity, it is great solvent to mix others when dealing with the polar compound. CDCCN, which is deuterated acetonitrile, this solvent has a relatively medium polarity comparing to others. This solvent has an affinity with water and is volatile, thus sample recovery is viable. CD2Cl2, which is the deuterated DCM. This is a more volatile analog of CDCl3 and is more solvent sensitive and react to the compound. Additionally, residual chemical shift in proton NMR is often ideal as it does not overlap with the signal with aromatic and aliphatic compound. Sample and solvent. The sample of 5 to 50 mg is dissolved in the solvent of 0.5 ml. Most commonly, CDCl3 D2O is placed in the sample tube, which is suspended to the magnetic field to set spinning. Ideal solvent, no proton, inexpensive, low boiling, non-polar, inert. Deuterated solvents used to eliminate proton signals from the solvent like CCl4, the CDCl3 and CS2. Reference compound. It is customary to measure the resonance frequency or signal of the individual nuclei related to the resonance frequency or signal from a reference compound. Universally accepted as a TMS. TMS is added to the sample as a reference and silicon is less electronegative than carbon. TMS is highly shielded and signal defines as zero. Organic protons absorb downfield to the TMS signal. Okay. So basically, since if NMR, in NMR we actually detect the protons and carbons also, so especially in proton NMR. So why CDCl3 is required? Because if we use normal chloroform which has proton, solvent amount is very, I mean greater than the compound itself, right? So the solvent peak will be so much higher that you will not see any compound peak. That is why CDCl3 is used when the deuterium is not NMR active. That is why it is used. But the CDCl3 that we use, or any deuterated solvent we use, it is not 100% pure. It is the 99% or 99.9% isotopic level. So that 0.101% of proton also shows a little peak in NMR. Okay, that is also a characteristic peak of a particular solvent. So remember that this is about the NMR spectra. So this was the last question of today's session. So today what we discussed, we mainly discussed about the secondary structure of the protein, along with we discussed our different techniques like of chromatographic techniques of the protein separation. And also we discussed about the enzyme kinetics a little bit. In the next class we will discuss regarding the enzyme kinetics in more details. I will be sharing my email ID here. So if you have any doubt or any query, please drop an email to it. So this is my email ID. If you have any doubt or any query, you can drop an email to it. Today it was our third live session for this NPTEL course of Organic Chemistry and Biology and Drug Development. I hope there is no issue. If there is any problem or any doubt, please let me know through this email. I will discuss that particular query in next class. Okay. Thank you for joining. Thank you for answering in the class. I will leave the session. Let's meet on the next Friday from 6 p.m. to 8 p.m. on our week 4 live session. Okay. Thank you. I will be closing the session for today. If there is any no issue. Okay, I am closing the session. Thank you all.